Tell us your name and tell us about your project.
Okay. Hi, my name is Maurice Wilkins. I am an English physicist. My project that I'll be discussing today is the use of X-ray diffraction to produce images of DNA molecules.
What were you guys trying to do or find?
So, like many other scientists post-World War II, we were all searching for a way to correctly map or model DNA strands. No one really knew whether the strands were a helix, if they were a helix, what type of helix, were there just single strands. So it was a lot of speculation during the time. So my group, we believed that using X-ray crystallography, we could begin showing the nucleic acids and proteins that make up DNA.
Who else was involved in the project?
So there were two other scientists involved. The other scientists were Rosalind Franklin. She was my colleague as well as an expert in the crystallography that we were doing, along with Raymond Gosling. He was a graduate student at the time, a PhD student. Both of us supervised him, and he worked very diligently in helping us get the research we needed. I also collaborated with James Watson and Francis Crick of Cavendish Laboratories. This was towards the end of our research, and I showed them some of our images that we collected, and they were very inspired by one in particular called Photo 51. In this photo is probably the best image that we could have had of the DNA, and from that I think it largely helped them with their discovery of DNA as well.
Where did the project or experiment occur?
So like I mentioned, I'm a London physicist, so the experiment itself took place at King's College in London. It was in a lab, a biophysics lab, that me and my colleagues were updating so that it would be able to be used for our project.
What other significant events were happening around the world at that time?
So our research really took root right after World War II ended, with the Allied powers winning. So there was a lot of uncertainty. On top of that, there was still a lot of tension and just smaller wars that were raging in other parts of the country or other countries in general. And all of it really made it hard for us as people, not just physicists, just people in general, to keep wanting to go on. So there was the Korean War, with North Korea attacking South Korea. There was the Cuban Revolution a few years later. There was a lot of tension between the Soviet Union at the time, because they had built their own version of nuclear, or the atomic bomb, and then right after that they ended up creating a treaty between them and China. Mind you, they were our allied powers in World War II, but having that power in communist hands wasn't very good for the rest of the world. But there were a lot of different things going on in the world at that time. There was also the North Atlantic Treaty Organization between some of the Allied powers and a few other countries trying to fight against the spread of communism. So all of this kind of accumulated and made it very turbulent for not just the United Kingdom, not just America. It was everywhere that people were feeling very stressed.
What was your role in the project?
So I think the best way to explain it is for me to start with my original research and then how it ties into this one. So I began working as a biophysicist in Scotland with my Cambridge mentor, John Randall. This was in 1945, and the following year we left because he got a professorship at King's College. So with this, I became a member of the Medical Research Council Biophysics Research Unit, and there I began studying the genetic effects of ultrasonics, but then shifted my studying to the orientation of nitrogen bases in DNA and RNA. So these would be your adenine, your thymine, your cytosine, your guanine, and uracil, which is an RNA. I also studied tobacco mosaic virus and the nucleic acids, and I noticed a uniformity or like a similarity in the general shape of all DNA fibers, and I wanted to further expand on this and see why that is. So I had experience with X-ray diffraction or crystallography from my graduate years at Cambridge, and from there I knew that if we could crystallize the DNA, it's much easier to diffract and get photos and images of the shape of the DNA itself. So I gave the PhD student, Raymond, I gave him DNA samples and asked him to diffract photos. So these images were very promising. The first few were nice. They made it easier and better for me to see that there were definitely similarities, and there was something right here that we could build on. So from there, we procured pure DNA samples, and from those we drew them into microfibers. And whenever we did the diffraction again, we found that these microfibers were crystalline like we thought. So that moved us into using the crystallography to further our research. So that's what my particular role was in that project.
What impact did your project have on the physics community?
So like I had mentioned earlier, the photos that I had showed to Watson and Crick, especially photo 51, these were photos that had, I don't know if I would say influenced, but largely backed what they were also considering, and that is that DNA is a double-stranded molecule, and that it wraps around itself in a helix form that repeats every at a consistent length. So these are things that before in the scientific community no one had backed, no one had physical evidence of, and we were one of the first people to actually have physical evidence from our crystallography and from the X-ray diffraction that proved these speculations. So the pictures that we were able to take greatly aided in the discovery and creation of the 3D double helix model presented by Watson and Crick, as well as it gave validity to the research that all of us had subsequently submitted in 1953.
With Rosalind Franklin passing in 1958 and you being recognized as aiding in Watson and Crick's discovery, do you believe that you and Franklin would have published your research on DNA and X-ray diffraction first had you worked better together?
I don't know truly if we would have. I would like to say that we would, but you'd have to understand that the relationship that Rosalind and I had was very strained, and this largely came from a misunderstanding that I would be leader over her, or vice versa, that she would be leader over me, and these were things that were not expressed with her being brought on board. These weren't things that were told to us, so it created a lot of tension in the early days of our research, and this eventually led to us not really speaking to each other and not collaborating as well as we should have. So I believe I believe that if we would have worked together, and if we wouldn't have been so caught up on one of us being supervisor or one of us being the superior over the other, we could have shared our information. We likely would have worked together so much easier, and there would be a very good chance that we could have published our work maybe a few months to maybe even years before Watson and Crick did. It also would have made it easier for our relationship as scientists to grow and to be more productive.